I study cell division, and I've been working on that problem for about 40 years. It's a fascinating problem because cells must divide in order to live. Cells are very complicated, so the division process is itself complex, because every time a cell divides, you basically have to build two new cells where you previously just had one. This means, of course, there's a lot of biosynthesis that must go on in order to provide the materials that will allow a cell to produce two viable daughters. Now, cells are very complex. Their complexity can be thought of in terms of the number of instructions that it takes in order to build a cell. And although we don't know that number precisely, you can estimate it from the number of genes and from the ways in which genes are regulated and from existing proteins in cell. And in short, the number of instructions that's involved to build a cell is more than the number of instructions that it takes to build even a very complex man-made object, such as a moon rocket or a supercomputer. This means that cells really are at the very high end of complexity in terms of the kinds of structures and systems that we think about. But on the other hand, cells are very small. They're so small that if you take cells from, say, the human liver, it takes something like 1 million of them to make an object as big as a pinhead. And this means they're not only highly specialized in terms of their complexity, but also in terms of their micro-miniaturization. Now, on top of this, cells can reproduce themselves. Imagine what you would have if you had a self-reproducing moon rocket or supercomputer. But cells can achieve this remarkable goal, and this is the process that we're going to look at in 3 lectures. If you look at this electron micrograph, you see, of course, the familiar nucleus, the cytoplasm with all its complex membranes. Not shown here are all the complexities of the cytoskeleton as well. All of these structures that we can see in a cell are actually made from the assembly of macromolecules, proteins in most cases, sometimes nucleic acids, carbohydrates, and lipids. And all of these constituents have to be synthesized before the cell is ready to go into division. So preparation for division involves a tremendous amount of biosynthesis. The instructions for this division come by and large from the DNA, which is located in the nucleus and has the familiar base pair structure that allows the sequence of the nucleic acids to define the sequence of a message, which in turn defines the translated product. And yet, that complexity of itself is not sufficient for building a cell, because you could take DNA and put it in a test tube and transcribe it and translate it, and you still won't build a cell. It requires a cell to build a cell. And so what we find in the structure of a cell is a template, in effect, which allows individual gene products as they're made to go ahead and assemble in place in order to build the structures that are necessary. Now, you can watch this process in phase microscopy looking at something like these 2 cells shown here, imaged in my lab a long time ago. It's a time-lapse movie which compresses about 20 hours of a cell cycle into about 30 seconds. So time is flying here, and you can watch any one individual cell, and as you watch it moving around, you'll see it divide. The periods between division, which are of course called interphase, are the times of synthesis that I've been talking about. It's then that proteins are made, RNA is made, DNA is replicated, so that we have 2 cells in one bag. The process of cell division, then, is the mechanics of separating all those constituents into 2 discrete objects. And it has to be done very well, because if cells lack essential constituents, for example, a chromosome, some of the DNA, which includes instructions for making RNA and protein, then the daughter cells will be unlikely to survive. So the division process is a very precise one that has to take advantage of structures that are built in order to assure the accurate segregation of the components of the cell, which are present in only a few numbers, like chromosomes. What you're seeing here is cell division going forward with no restraint. The cells are being provided with the factors that stimulate their division, they're being provided with all the food that they need, and so they simply divide as rapidly as they can complete the essential synthetic processes. This is like the lifestyle of a unicellular organism, where food is really the limiting factor in how a cell can go forward, making decisions to divide and produce two of itself. For the cells in our body, on the other hand, this process must be tightly regulated, because if cells divide too frequently, you can get anomalies which are very dangerous medically, like cancer. On the other hand, cells must divide in order to achieve a healthy adult organism. Of course, the cells of an embryo divide in order to produce the juvenile and then the adult form. Cell division is essential in wound healing. Cell division is also essential in tissue renewal. So, for example, the red blood cells that circulate through your vasculature are comparatively short in their lifetime, a couple of weeks, and if they are not replaced, you will have anemia. So cell division is being regulated in a multicellular organism to produce all the cells that are necessary to balance the cell death which is occurring.